Welcome to the 8th unit of your science textbook, that is about characteristics of organisms. First we will see what are the common characteristics of living organisms. Actually you have learned these common characteristics in your lower grades also, but again we will see what are the common and major living characteristics of organisms. So the first one is cellular level organization, and then nutrition. Third feature is the respiration, then irritability or coordination, excretion, movement, reproduction, and growth and development. So these are the main characteristics of living organisms. Now we will study these characteristics one by one. So the first characteristic is cellular organization. Now in the unit 6 you have learned that all living organisms are made up of cells, and cell is the basic structural and functional unit of the living organisms. So if you consider organisms, there are two main types of organisms as multicellular organisms and unicellular organisms. As you know, unicellular organisms are made up of only one cell, and all the metabolic functions are taken place within this one cell. And if you consider the multicellular organisms, they are made up of two or more cells. So multicellular organisms are in two main types as plants and animals. Now in the topic of cellular organization, you are mainly going to learn about the multicellular organisms, that means about the cellular organization in plants and animals. Now you know that single-celled organisms are made up of only one cell, and they are having an organelle level organization. But if you consider a multicellular organism, they have a different level of organization. So this diagram shows the level of organization that you can observe in a plant or in an animal. So cell is the basic unit of living organisms, and by combining cells it forms different tissues, and these tissues get together to form different organs which perform various functions, and these organs get together to form different systems, and these systems unite to form one organism. So in a multicellular organism you see a tissue or organ level organization. So this is the basic levels you see in an organism when you consider their organizational structure. Now we'll see how this cellular organization or cellular level of organization can be identified in both plants and animals. For example, we'll take the embryonic development of human. Here in fertilization, an egg cell and a sperm cell combine to form the zygote, and by further cell division it forms an embryo, and this embryo develops into the multicellular organism by further cell division and the development of cells. So the same phenomenon is occur in plants. There are also a pollen grain and an ovule fertilized to form an embryo, and it further develops into a plant. So in these two instances you can clearly identify that the cellular level organization can be observed in the development of both plants and animals. Now we will move to the next living feature of organisms, that is nutrition. So the definition for nutrition is that it is the process of taking food in and converting it into the energy, and this energy is used for cell growth and development in all organisms, and usually the nutrients act as the provider of energy for all organisms. And if you consider the way of obtaining energy to the organism, there are two main types of organisms as autotrophic organisms and heterotrophic organisms. As you know, autotrophic organisms are the organisms that produce their own food by themselves. But heterotrophic organisms cannot produce their own food by themselves, and they depend on the food produced by autotrophic organisms directly or indirectly. So these food relationships you find among organisms can be clearly identified by studying food webs and food chains. So this theory is given to you in your lower grades also, but here you are going to learn more about the nutrition of organisms. When you consider the autotrophic organisms, you know that autotrophic organisms can produce their own food by themselves. So autotrophic organisms are usually in two main types based on the type of energy that they are using for producing food. The first type is photoautotrophic. Photoautotrophic organisms means plants or the photosynthetic organisms. So you know what is photosynthesis, and in photosynthesis these photoautotrophic organisms produce food by taking solar energy as the source of energy in food production. So for example, higher plants like all the trees you find in your environment comes under the group of higher plants, and euglena, green algae, and bacteria species like cyanobacteria also come under the group of photoautotrophic organisms. So the next group of autotrophic organism is chemoautographs. So this is a new term for you. Chemoautotrophic organisms means the organisms that produce their own food by using the energy formed by the chemical reactions with organic molecules. So the energy produced by these chemical reactions are used as the source of energy in food production of these organisms. For example, sulfur oxidizing bacteria, nitrogen fixing bacteria like cyanobacteria and rhizobium, and iron oxidizing bacteria is also a form of chemoautotrophic organism you find in your environment. So according to the type of energy used by the organisms in food production, there are two main types as autotrophic and chemoautotrophic. Now we will move to the next characteristics of living organism, that is respiration. So as a living characteristic, respiration is not taking air in and passing it out of the body. Actually, the respiration is a process by which the stored food is transformed into energy inside the cells of an organism. So here the stored food means the food that is absorbed into your circulatory system by digestion, and this food is stored in the storage tissues of organisms, and it is burnt with the inhaled oxygen and produce energy in the form of ATP. So this whole process is known as the respiration in organisms, and this respiration process is taken place within both plants and animals, but there's no special way to observe this process. So you can't observe cellular respiration by experiments, and you can only observe it through the respiratory movements of the organism. So you have learned about these things in your lower grades, so I am not going to explain it further. And here you are going to do a few experiments to find the respiration of organisms. So now you all know that the oxygen is taken into the body by inhale, and that inhaled oxygen is used in producing energy by the process of cellular respiration, and as a byproduct of cellular respiration it produces the gas carbon dioxide. So this carbon dioxide is a waste product produced in your body, so it has to be released from the body. So that carbon dioxide is released by exhaling. So the only thing you can do to identify the process of respiration is to find that the oxygen is taken into the body and carbon dioxide is released out of the body during the process of respiration. So here you are going to do two more experiments to find whether the oxygen is absorbed or inhaled into the body during respiration, and carbon dioxide is released from the body during respiration. First, I am going to discuss with you about an experiment to show that oxygen gas is absorbed during the process of respiration. For doing the activity, you have to prepare two setups as shown in the diagram. And the setup A is the experimental setup and setup B is the control setup. So we are using a control setup in an experiment to confirm the result we obtained at the end of an experiment. So before starting to explain about the experiment, I have to tell you something about this potassium hydroxide or KOH. KOH or potassium hydroxide is a chemical that is used to absorb carbon dioxide in the environment. So now we will move to the experiment. So as you prepare this setup, you have to seal the conical flask using this cork lid. So as you seal the conical flask using cork lid, you can confirm that there is no gaseous exchange between this conical flask and the environment. So we have put germinating seeds into the conical flask, and these germinating seeds perform respiration. As it is doing respiration, it absorbs oxygen from the gas containing in the conical flask and releases carbon dioxide into the conical flask. That released carbon dioxide is absorbed by this potassium hydroxide. So it creates a gap in the air containing in the conical flask as oxygen is absorbed by germinating seeds and the released carbon dioxide is absorbed by potassium hydroxide. So the air containing in this U-tube moves gradually into the conical flask. As the air moves into the conical flask, the water level in the U-tube rises up to fill the air gap. So as you are doing the activity, you will observe that the water level of this U-tube rises up. So this rising of water level is occur due to loss of gases or due to creating a gap in this conical flask. So that gap is created as the oxygen is removed from the conical flask for respiration by these germinating seeds. So, but you cannot observe any change in water level in this U-tube of setup B because we have not applied potassium hydroxide in this setup. So as the oxygen is absorbed by these germinating seeds, it releases carbon dioxide also. So that released carbon dioxide fill the air gap that is created in this setup. So there is no rise of water level in the setup B. So here are the assumptions we made while doing this experiment. The first assumption is that the volume of carbon dioxide in the flask is negligible, and next you have to assume that the volume of oxygen that is absorbed is equal to the volume of the carbon dioxide that is released during the process of respiration. If it is not so, we will not be able to conclude that the rise of water level is occur due to the loss of oxygen. Now we will move to the next experiment, that is to show that carbon dioxide is released during respiration. Here you have to make somewhat complex experimental setup as shown in this diagram. and also you have to use a live specimen in the experiment. So here in this diagram it has used a rat, but in your textbook the experiment is explained by using a frog instead of this rat. However, you have to use a live specimen to perform this experiment. Before starting about the experiment, I will say what are the things that is contained in each these conical flasks. In this first setup or setup A, it contains potassium hydroxide, and now you know the purpose of using potassium hydroxide. It is used to absorb carbon dioxide in air. And here in this setup B, it has used lime water. So we use lime water to identify carbon dioxide gas. In the presence of carbon dioxide gas, this lime water turned milky color, and you have done this experiment also in your lower grades, so I think you can remember the color change. And here in this specimen C, we have used a live specimen. Here it is used a rat, and in the setup D again it has used lime water, and in setup A the bottle is filled with water and it is connected a suction tube or a tube that is used to suck the water inside the bottle. So now I am going to explain how the experiment is going to be done. As the setup is prepared like this, you have to suck the water from this suction tube. The suction tube is connected to bottle E. So as you suck the water from this suction tube, the water level of this bottle goes down and it created a air gap. So to fill that air gap, air inside these other bottles going to move into bottle E. So it created an air flow from bottle A to bottle E. So as the air enters to this setup A, carbon dioxide in this entered air is absorbed by this potassium hydroxide. So the air leaving this setup A or bottle A does not contain carbon dioxide and it is free from carbon dioxide. So that air is entered to this bottle B, and here it contains lime water. So now you know lime water is used to identify carbon dioxide, and in the presence of carbon dioxide, lime water turns milky color. So here you can observe no color change in lime water as the air does not contain carbon dioxide. So air free of carbon dioxide is entered to this bottle C that is containing the specimen. So this organism inhaled the air in the bottle and it exhaled carbon dioxide into the bottle. So that air leaving this bottle again containing carbon dioxide as this organism releases carbon dioxide by exhaling. So this bottle D or conical flask D contain lime water. Here you can observe a color change in lime water, and this colorless liquid turn into milky color because the coming air containing carbon dioxide. That carbon dioxide is from the exhaled gas from this organism. So by this experiment you can clearly confirm that carbon dioxide is released as a byproduct during respiration. So performing this experiment we have used a rat, and instead you can use some other animal like frog, or either you can use germinating seeds, or for germinating seeds you can use germinating gram or germinating green grams, paddy seeds or germinating bean seeds instead of using an animal. So that is about the respiration. And Today I did 3 characteristics of living organisms, that is about cellular organization, nutrition and respiration. So in my next video I am going to discuss with you about the other living characteristics of organisms. Goodbye you all.